At the Soho 2020 Italy meeting, my presentation was to discuss the current role of antibody-drug conjugates and bispecific antibodies in multiple myeloma. So this represents some of the most exciting classes of drugs being developed for the treatment of multiple myeloma currently. So antibody-drug conjugates are particularly relevant given the recent approval of Belantamab Mafodotin, which is a first-in-class BCMA antibody-drug conjugate. I think some of the highlights of this particular drug is that although the single-agent activity was fairly similar to other recent single-agent approvals in multiple myeloma, the depth of response and the durability response of 11 months at the 2.5 milligram per kilogram dose was particularly encouraging. And so this will represent an exciting off-the-shelf approach to targeting triple-class refractory multiple myeloma, which is really an unmet need currently in multiple myeloma therapeutics today. And it's important to note that, because of the ocular adverse events that are seen with Belantamab Mafodotin, it's important to manage patients in a multidisciplinary approach with eye care specialists to effectively and safely dose patients who are receiving Belantamab. The second part of my talk was about bispecific antibody approaches. So this is also a very exciting off-the-shelf approach for treating multiple myeloma patients. So most of the bispecific antibody approaches have been targeting BCMA as the tumor-specific antigen and T cells or CD3 on the T cells as the immune effector of choice, cell of choice. And so this basically brings a T cell and a myeloma cell expressing BCMA in close proximity, leading to myeloma cell death. So in my presentation, I discussed several bispecific antibodies that have had clinical data reported to date, including AMG 420, including CC93269, and Teclistamab, which was recently reported at ASCO this year. And so in summary, I think bispecific antibody directed approaches are very exciting in multiple myeloma. Again, they're off the shelf, which could confer some advantages over autologous CAR T cell products, given that patients can be immediately dosed, particularly those that are rapidly progressing. They generally are associated with some degree of CRS, although severe CRS or neurotoxicity has not been reported as much with the bispecific antibodies. And I think, again, these approaches will be very exciting to see how they mature in the myeloma clinical development space.